I hope I'm audible and everyone is settled in. Hi everyone. Biorix is a student-run biology community built for curious minds across the life sciences, from genetics and microbiology to ecology, zoology, botany, and conservation. It serves as a collaborative platform where students connect, exchange ideas, and engage in meaningful scientific discussions. As part of our exploration of biodiversity and conservation science, today's session focuses on ecological niche specificity and the conservation of threatened plant species in the Western Ghats. Today's session is titled Ecological Conserving Threatened Low Elevation Trees. To lead this session, we have Dr. Devika M Anilkumar with us. Dr. Devika is an ecologist and conservation biologist currently serving as a postdoctoral fellow at BGCI UK and research associate at the Western Ghats Hornbill Foundation. Her work focuses on plant biodiversity conservation, ecological niche modeling, habitat restoration, and the conservation of threatened plant species in Western Ghats. She has contributed extensively to IUCN Red List assessments, authored several research papers, and received recognitions including Ruford Small Grants and the Kerala State Biodiversity Board Doctoral Fellowship. Her doctoral research at the University of Calicut focuses on the niche specificity and overlap of threatened angiosperms and their application in conservation and eco-restoration. We are delighted to have her with us today and look forward to an insightful session. Let us warmly welcome Dr. Devika M Anilkumar. Ma'am, over to you.
Thank you, Rosemary, for introducing me. I hope I'm audible to you all. Now, I think I can move on to the session. Hope my slides are visible to you, right? So today I'm here to share a few insights on ecological niche specificity, eco-restoration, and as well as a framework for conserving the threatened low elevation tree species, especially in reference to the Western Ghats region. So the practice of conservation within the framework of climate change has become increasingly intricate as various species, ecosystems, and the entire landscape due to the shift and the alterations to the species composition in response to the changing climate has become very critical, and to the because of this climatic conditions and the shift in the climatic situations, we have to carefully look upon how our ecological restoration practices has to be unique and ecosystem based. So the conventional, conventional conservation strategies are now being augmented and redefined. So the climate change linked policies has to be unique and as well as ecosystem based. So the UN declared this decade as decade of ecological restoration, and lots of ecological restoration practices are ongoing from local to the landscapes to the global level too. So in 2004, I think it is SER, that is the ecological, SER group defined eco-restoration as the restoration or the recovery of an ecosystem that has been degraded, damaged, or destroyed. So when we look upon the ecological restoration, the biopropagation or the planting of a single species can be lead to the invasions to the other niches. And as well as the eco-restoration is sprouting everywhere, and but actually most of the practices lack a minimum protocol to ensure that it is differ and different from the traditional plantation practices. So when we look upon the global IUCN Red List status of the threatened species, because I share this slide due to when we look upon the conservation of an endangered species or recovery of a threatened species or whatever, we should be aware about what is the current status of that species or what is the current status of that tree species, and as well as the associated species that we are planting for the restoration. So in the current global red list status, more than 48,600 species are at the brink of extinction, and 28 need to be still assessed. And nearly 1 lakh species are being assessed, and their global goal is for 260,000 species for assessing yearly. So we need to know, or we should be concerned about the status of the species while we are planning for the recovery of that species. So we should know what is the population status, or what is the population that the species currently have, or what is the distribution status of that species, and that we can gain through collecting the occurrence records, global databases, and even from the herbarium reports. So we should scrutinize all these things, and from that we can do the IUCN Red List assessment too. So herbarium review is one of the critical aspect while we do the IUCN Red List assessment. And when we look upon the status of an individual species, different categories were there in the red list, and we usually do the red list assessment based on the IUCN guideline, and this is the latest guideline which is recently released in March 2024, and different criteria and categories are there for the assessment. And our team has assessed some of the low elevation tree species in the Western Ghats. Well, one among that species is the low elevation tree. That is one is here, that is Rasparis criminata, which was assessed as critically endangered because earlier in 1998 and 1999 period, the IUCN Red List assessment was done only based upon the criteria B, that is the extent of occurrence and the area of occupancy. So it is actually, there was only limited data for each and every individual species. But the reason the assessment we look upon, we can understand that the multiple criteria were incorporated in the recent assessment. So that is one of the major difference between the earlier and the recent assessments. So if we could able to incorporate the more population data and ecological data, we will get a fine-tuned ecological threatened status for that species that we are trying to assess. So when we do the assessment of these species, we incorporated both its population data, number of mature individuals, and as well as its ecological data. Earlier, this Rasparis criminata was endangered, but when we incorporated the population data and the species has severely fragmented distribution, the species comes under critically endangered. But recently, more publications are and reports from Karnataka is there. So there will be a change and shift from the category for this species. And the next species, that is the Prioria pinnata. This is also one of the low elevation tree species that we can see in our Western Ghats, where this species was also endangered earlier, and we could able to do its reassessment in recent year, and were assessed the species into vulnerable category. So these are a few low elevation tree species that we can see. Apart from this, there are lots of associates like for the species, Dipterocarpus indicus, Hypnocarpus pentandrus, and all. So when we look upon the ecological restoration of an individual species, or for the recovery of a species, the threatened status is one of the critical matter that we have to look. And we all are now familiar with the ecological niche modeling tool, which is nowadays used for the potential habitat prediction of the species. By incorporating the bioclimatic and vegetation data too, we can refine the potential habitat of a species. So actually, the ecological niche modeling were done. We can do both in R and manually using the QGIS software and the Maxim software. And the current prediction and as well as the future climatic habitat prediction is one of the critical tools that we can use for the potential habitat prediction of our targeted species. And so the understanding the landscape is one of the critical one that we want to look upon, even what is happening in the current situation and what is happening in the future situations for the targeted species that we are trying to restore in the landscape that we are looking on. So both this current and future habitat status or habitat suitability prediction we can do through the ecological niche modeling, and our restoration has to be ecosystem based, niche specific, and it should consider the heterogenic composition too. So while we are planting an individual species, we should plant its associated species too while we are doing the restoration of that particular species. And we have developed a methodology for the conservation of this threatened species in the Western Ghats. Few are published in elsewhere, and one is that species is Cryptocardia and Maliana, and we developed the niche profile and niche model that is for the species, and also the other species that we come across that we developed. The potential habitat suitability map for the species like Thyrea pinnata, Syzygium caryophyllatum, and all these were published recently in a few journals. And here I'm showing an example in the case of this Diosperus culminatus species, that is one of the low elevation tree species, which is a critically endangered species. When we look upon the bioclimatic prediction that using the Maxim software, this is the actual distribution of that species. This green color dots is actually the actual distribution of the species, and this map is showing the potential habitat is suitable for the ecological restoration for the species. This is actually based upon the bioclimatic factors, that is the environmental factors, which is derived from the WorldClim, and it's freely available in the WorldClim. You can download the bioclimatic layers, and you can predict the potential habitat based on the bioclimatic variables. And this resulted when we done this analysis, which resulted the temperature, seasonality, precipitation of the wettest month, and here these are the bioclimatic variables that shown the major suitability or the species that the bioclimatic variables that contributed to this species, and also the non-bioclimatic variables like slope and elevations is also showing some of the contribution to this species. And this is the jackknife map which showing the average contribution that is for the species, which is an output from the Maxim. And one of the limitation of this ecological niche modeling habitat suitability based upon the Maxim is that it is only confined to the bioclimatic factors. They're not incorporating the vegetation factors or the terrain features. So to overcome the limitations of the the niche modeling through the Maxim, that is, we can say that it is interpreting the Grenelian niche, that is the niche which is based upon the environmental factors. So that to overcome that limitations, we can incorporate the vegetation layers, and we incorporated the vegetation layers and the terrain features, and we could able to refine its prediction like this. You can see this green color indicates the areas which is having the conservation potential, and as well as the restoration potential, and this black color indicates the converted forest areas, and the orange color indicates the areas having the non-forest converted regions. So by incorporating these terrain and the vegetation layers, we will get the conservation areas, ecorestoration areas, and converted forest habitat for the species. So based on this, we can plan which area that we can we can consider for the ecological restoration for the species. And another example I'm showing in the case of this Thyrea pinnata. See, this is the actual distribution of the species, and we got this prediction like this. This red color indicates the areas having the higher habitat suitability, and the blue, which is leading to the low suitability in the blue color. And see, by incorporating the vegetation layers, the area get refined into this like this, and the major conservation area that we could able to locate in the ecoregions like Coorg, Cormoran, Varshnad regions. Actually, this is the ecoregions which is based upon the same ecoregion layer. And another thing that we want to look upon is the Eltonia niche factors, that is the species interaction factors. So for that, we developed a niche profile based upon the is 0.1 hectare square plot for that species. And this is an example I'm showing in the case of a media elevation species, that is the Cryptocarya anmaliana, which is we published in the elsewhere, and you can freely available that. And also this Cryptocarya anmaliana is a critically endangered species, and this is the niche profile that we developed for this species, where we can see which all are its associated species, and through that we can understand which composition that we need to be planted while we restoring the species, and why trees matter rather than any other herbs or shrubs in an ecosystem. Actually, the trees can serve as a keystone role, and these supports the growth of other biota in that ecosystems, and the trees, when we look upon or focus on the ecological restoration of the tree species, we can ensure the growth and the support, or the support and the growth for the other herbs, shrubs, and all other biota in that ecosystem. So now I am showing an example that we done in the one of the low elevation region in our Kerala, and one model that we developed for the Diosperus criminata. First, we assessed the species, where the species was published in the IUCN Red List. Actually, the species had 6 subpopulations of 200 to 420 mature individuals, and which is available in the IUCN website. So, and we identified its population, and we predicted the potential habitat for the species, and this was the predicted potential regions. See, the Greenland niche shows a higher prediction for the species. You can see that, but we see its actual prediction, it is too much limited, which is actually based upon these species are dependent on their Eltonian factors, that is the species interaction factors, which is actually controlling the distribution of the species. See, when we observed or monitor the species for 4 to 5 years in the field, the role of seed dispersal agents was very critical, because its fruits were eaten by the other frugivores, hornbills, and all. And this is the Eltonian niche, that is, we can say that the niche profile that we developed for the Diosperus criminata species, and these are its associates, and this is one of the 0.1 hectare square plot representation for this species. So we can use these as the model for ecorestoration plot, and we can say that the species not exist as an individual, because it is exist along with its community. So when we planning the restoration of an individual species, we should plan its associated species to along with it, which can ensure the success and the survival of that species, and as well as the greater success rate in that ecorestoration process. And we later we collected the seeds and saplings, and we developed germinated the associated species too for the ecological restoration of the species. And the major associates of the species, which include Dipterogarbus indicus, Priore, Pineta, Hopia parviflora, Hopia ponga. So these saplings were germinated, and the one of the biotic interaction and the proper phenological event were found, which is essential for the germination of this species because it's having an association with the Cyaneria fungi too. And we germinated 200 saplings first and later into 500, and the saplings were produced more in our nursery, and which is used for the specific restoration model in the forest regions and as well as the low elevation identified site. So this was the restoration model that we developed, and we identified the potential ecological restoration sites based upon the habitat predicted through ecological niche modeling and the refined vegetation layer incorporation. So these are the major steps that we have identified. First we should identify the potential area, then later we should look upon the ground truthing factors, and from that we can identify which all areas that we can plant the targeted species along with its associates. And another factor is critical is the stakeholder engagement. The stakeholder engagement is one of the another important factor, and by incorporating all these features, we identified a few of the selected sacred groves in our region, low elevation region, which is having a resemblance in the low elevation evergreen forest too. So we first we have done the ground truthing, and we noted the associated species which are lacking there, and we planted these species along with the associated species in the identified low elevation sacred groves. This is one we done in the Srinarayanagudam Panchayat, where we done first we prepare a detailed plan and identify the ecosystems, and we planted these species in the identified low elevation sacred groves, and this was funded by a Pragati Research Fellowship. And like this, we planted these species and associates along with the identified sacred groves in this region, and which actually this was done by incorporating the MGNRG scheme. So the woman is ready to work with us, and we collaborated with the BMC Biodiversity Management Committees of the Panchayat, and the support of the Kerala State Biodiversity Board was also critical. And through this convergence mode, we collaborated with all of them, and we planted the selected species and the targeted species along with the associated species in the identified sacred groves in this region. And this was a sapling growth and survival that we observed for these species and its associates, and nearly 89% survival growth were observed, and the between 1 and 1.5 year of monitoring data is shown here. So the community is working with us, is ready to monitor. So we could able to ensure the monitoring and as well as the success rate of the planet saplings in that region. Actually, they were also doing the working and collaborating with us in the coastal restoration, coastal vegetation belt restoration in the 4 different wards in the Srinarayanagudam Grama Panchayat. And so we could able to easily accommodate them in the restoration of the sacred groves too in that identified region, and they were ready to plant. And this was one of the success stories that I'm showing in the case of the coastal region. And our MGNRG woman working in them is still continuing monitoring and as well as watering on these planet saplings in the site. And for this convergence mode model, this Panchayat, the Srinarayanagudam Panchayat, that is the BMC, Biodiversity Management Committee of this Panchayat, received the Best BMC Award from the Kerala State Biodiversity Board last year. So I was trying to point out that when we plan in a restoration model, it should be in a convergence mode. So from that, we can ensure the sustainability and as well as the success and the survival rate of the planted saplings. And through this process, we can ensure the involvement of the local community and as well as a stakeholder involvement. And through the, through this project, or through this process, we can ensure the sustainable developmental goals through the women who are working in this restoration project can ensure no poverty, because the women working in this will get 100 days of working days, so they can meet 100 days working days for the women who are working in this restoration project. But the other NGNREG women won't get this 100 days complete, because they won't be working days for them and working mandate for them. So this actually the women working in this our restoration project was ready to take over this work, and we could able to ensure no poverty to them, and also zero hunger, and the students who are there in the nearby institutions can ensure the quality educations, and as well as we can ensure the 13th SDG, that is the climate action and the life on land. So when we plan an ecological restoration or a recovery of an individual species or an ecosystem, we should develop a framework through this convergence mode, and which can ensure the more and more sustainable goals too. That was to point out. And I just want to say that one more thing. When we incorporate this our women and as well as the policy makers and government, we can ensure this methodologies sustainability and survival, and this methodology was also implemented in the virtual forest division. That is actually the one of the real habitat for this targeted species, that is Diosperus pulmonata and as well as Prioria pinnata and its associated targeted species. So from there also we involved the Carter indigenous community and the Forest Department collaboration for the ecological restoration of these threatened species in the identified degraded sites in the virtual forest division, and the nurseries, the community's nurseries was also maintained along with them in the virtual forest division. So from that, we can increase the success of the ecological restoration, and as well as through this, we can maintain a convergence model for each and every species that at the brink of the extinction. Another thing I would like to say that if we develop an plan by incorporating the Grenelian niche and as well as the Eltonian niche factors, that is the Grenelian niche is the which only confined to the environmental factors, which will only we will get through the Maxim modeling. So by incorporating the terrain features and the vegetation layers, we can fine tune the predicted layer or the predicted area from the Maxim model into a better one, because our vegetation, we should know what is actually happening in our ground truth level. So if we done the phytosociological sampling in the identified site too, we can incorporate more and more biotic factors. So when we plan the ecological restoration of the species, the incorporation of both these Grenelian and Eltonian niche factors are too much critical, which can actually ensure the survival and the success of this identified plant species restoration. And another thing I wish to say that when we plant an individual species, it can become the intrusion to the other species niches too. So identifying the proper habitat is too much critical. We can't plan then we are planting lots of species in the June 5th without looking upon what is their niche, what is their habitat and all. So if we focus on the conservation of a threatened species, we can't plant a low elevation species in the high elevation species. That is actually doing an intrusion or the invasions to the niches of the other species too. Sometimes maybe the species may survive there, but will be an intrusion to the niches of the other species. So we should be aware about, or at least we should have a framework before planting each and every species in a large, huge number in an ecorestoration process. So by incorporating this methodology and the involvement of the community, we can maintain the monitoring and the survival of these saplings too. So I would like to conclude with this, and before that, I thank my funders. Ruford Nature Conservation Grant from 2022 onwards, Ruford is funding for me. So I thank Ruford and IUCN Red List and BGCI for the collaboration for the IUCN Red List assessment. And I thank Hornbill Foundation and Kerala State Biodiversity Board and Prakriti Research Fellowship, which actually contributed for the restoration of these low elevation tree species in the sacred groves. And also I thank the SER and other collaborators at this time. And thank you once again. You may ask questions if you have anything related to this.
Hello, Devika. Can you hear me?
Yes, sir, I can hear you.
Yeah. One of the questions is that what was the factor which made these trees very low in number?
Sir, your audio is not that much clear. Can you please repeat that question?
What was the precise reason why the population of these trees got declined?
Not audible.
What was the precise reason why the population size of these trees became very low?
Oh, the reason. Actually, each and every species have different reasons. In the context of our Western Ghats region, these low elevation species are influenced by the impact of the dams, reservoirs, and the plantations. These are the major factors or the major threats that affect the distribution of these species. So when we do the IUCN Red List assessment, we should able to see the past reduction for the species, because due to the plantations that we have implemented here in this landscape was one of the major threat for the species. But in the case of the low elevation species, the dams and their regulation is one of the other factors, and the infrastructure development, we can say all these factors are one of the major threats for the species.
I was asking about specific species, specific factors like diseases. Have you observed any?
Yeah, in the case of the Diospyros criminata, actually, the species is facing very poor regeneration and germination status, because it's having a very higher seed coat for this species, and it won't germinate in the natural habitat properly. So when we observe these species, we observed one interesting factor, that the Silaria fungi is having an association with the fallen fruits for this Diospyros fruits, and this actually they are trying to break down the seed coat of this Diospyros criminata seeds, which actually supporting them to germinate in the natural habitat. But even though we observed very low germination rate in their natural habitat, so we want to look upon the other propagation methods for these species. That is one factor that we observe.
So that essentially means that even if you plant more trees, you're also going to face the same problem, or else you will never be able to restore the species to the same population level, because it doesn't run by itself. It is impossible for us to replace the trees for thousands. Essentially, whenever reproductive success is arrested, that tree is going to be extinct, and you cannot do anything about it. Even if you increase its number, those things are also going to same thing. So that is what we call a distinction. Not all trees are trying to leave. There are some go. Uh, on the other hand, the first reason we should do, if it is just the habitat destruction, those plants are restored. But any tree is facing a reproductive failure can never be restored. So that's my feeling. Even though if you increase this number by having saplings, they are going to face the same problem in the future, and they are also going to decline.
Sir, can I add one more point to this?
Sure.
Another thing we can say that these species is also having the distribution which is dependent on the dispersal agents, because hornbills, civets are eating their fruits, and from their scat we can observe the germinated germinated seedlings of these trees. So if we could able to increase the number of this, targeted species, even if it is diascaros or any threatened species, along with it associate, we are not targeting an individual species. We are building up an ecosystem or a community. So it will directly or indirectly supports all the biota in that ecosystem, which will able to manage the all the interactions between each other. So if we could able to develop an better ecosystem, so the actual biotic interactions will increases automatically. So I think they will be able to cope up their germination low rate with the dispersal agents, because this better exchange will happen, and more seeds will germinate, I think so.
That we need more data to predict that. We need much more data which is going to be successful or not. So that's the first thing. The second about which you have done. Whenever you use a Berlin data, the problem is that it is basically extrapolated information from the available stations. So it will give that, but the problem is that it is very good weather station. And in the forested area have very, very little distribution of weather stations. So we cannot depend upon Berkeley data basically because it is purely extrapolated. The better way is to do it yourself, looking at the making our own measurements or measurements from the nearest weather station. And you can also do your own computation. Like for example, you can do measurement at one specific habitat. At the same time, you can compare it with the nearest weather station, and you can derive a formula for the correction factor. And that is the way it has to be done, because working data is just pseudo data.
Sir, actually, in this methodology, we are not only depending on the virtual data, because the ecological niche modeling, we can say like that, because that is actually the limitation of that methodology too, because it is only confined to the bioclimatic layers or the environmental factors, and at the same time we are trying to overcome the limitation of that Maxon modeling by incorporating the terrain features and the vegetation layers, and by doing the ground truthing and the phytosocial sampling, and also by incorporating the the population data and the herbarium through the herbarium scrutiny, and also all these are not virtual data, no. So this is actually through the plots we developed through the phytosocial sampling. We could able to fine tune the predicted area into a better one. That was we are trying to prove here.
Yeah, understood. What I mean to say is that even if you delete the math and modeling part, there's nothing going to happen with your argument because that is just not needed, because the other data which we are collecting, starting from the terrain data, vegetation data, and the ground truthing, that is quite enough for doing this sort of a study. Even if the virtual data is not at all used, nothing is going to happen to your study. That's my feel. But thank you.
Sir,
Thank you very much for the session.
yeah, we can. I just want to add one more point. Through this Maxent modeling, we can predict the current and as well as the future. Future. We can't know what's happening in future too. So through
But
this,
that is no, since the data is not real, that prediction is not going to be true. That's the problem. Because that information is derived based on absence information, which is extrapolated from far away weather stations. So that is the big limitation. So any prediction based on that data is not going to be far away from the reality.
but
That's the
here we
problem.
are using the presence only data also, so we can manage the absence. No.
No, but then basically Maxent relies a lot on the worklimb information on humidity,
Yeah,
temperature variations,
that
things
is
like
right.
that.
Very
But then
right.
that
Because
data is
actually
entirely that data is not true. That's the problem. So I personally feel that in your study, that's a very interesting study. The most important thing is that we are not just focusing on an individual species, but all the associated species. That's a wonderful thing which you have done. And data itself is quite good. So I personally feel that there is no need to be derailed based on Maxent information which is provided by Maxent. There's absolutely no need to rely on that. But thank you. Interesting work. Best wishes,
thank
Devika.
you.
Hello? Hello?
Yeah, yes, sir. You're audible.
Ravi from Palakkad. Your concept can be applied all across the different vegetations of Kerala, to climatic and soil difference of Kerala?
Yeah, yes, sir. Actually, the Maxent modeling is for the global databases, and the vegetation layer that we used is for the Western Ghats region. So we can use this methodology for the Kerala part too.
Okay. But this plants and all, can you use the traditional plants of the area?
Yeah, definitely. The native plants that we collected, the seeds from the forest, and we germinated its saplings and like that. Native plants of India, which is endemic to the Western Ghats.
Okay. Okay. Thank you. Thank you very much. Thank you very much. Thank you. Thank you.
So you have took two sampling like control or treatment, or only you have took the sample in like human modified landscape. Where you have collected this data?
Sir, can you please repeat the question?
You have sampled in natural habitat only, or like human controlled environment. Means control or treatment, both group or only treatment group.
Oh, yeah. Mm, actually, my PhD study was on the 25 species red list assessment and the ecological restoration plan development. I just shown only 2 species cases here, and actually I could able to collect the data for 126 plots and samples in the entire and Western Ghats landscape, and I just only shown here for the 2 species case. That's why I think you got this question.
But during sampling condition, microclimate factor is more impact during their phenological cycle, not phenological cycle, you can say during their growth development, microclimate factor, and that period is the more mitosis going on. So you are not thinking how this microclimatic factor, how you will predicting model using this remote sensing.
Sir, we can predict a species habitat based upon the bioclimatic factors through ecological niche modeling by using the Maxent. But when we look upon the ecorestoration or species recovery of an individual species, um, it is a mandate, or it is very critical that we should look upon the Eltonian factors, that is the species interaction factors. So for that, at least a minimum of samples and population data has to be collected for the targeted species that we are trying to restore, and that I have done for my study, for my PhD study, where I could able to collect the samples data from 126 plots, and based upon that. Actually, we identified the potential habitat for the targeted species, and if we could able to locate a species, like this is the potential habitat, but we should do the ground truthing, and we should collect the first minimum baseline data, what through based upon at least SER methodology, and all we can adopt that. But ground truthing is one of the mandate where when we look upon the restoration action. So through that we can collect the micro level baseline data too.
Okay, but you have took the treatment sampling. So how will analyze this data, that habitat modeling in your max extent JS?
Oh, I think, yeah. We can incorporate the vegetation layers from Singh et al. Singh layers that is available in India Biodiversity Portal and other sites too. So by incorporating these vegetation layers, even if it is by overlaying with the vegetation layers, we can understand that whether the identified site is a plantation or a degraded site or an shola or a primary or secondary forest. By incorporating these layers, we could able to find out that what is actually this identified site is it. So this is we can identify through a spatial manner only. So, but we can only ensure it through the ground truthing. So we need to do first first we want to use this vegetation layers incorporation, and after that we need to do the phytosocial sampling in the identified site, and then incorporate this microclimate layer. Just it is not incorporating with the maxent data or something like that. We can only use the the biotic layers like elevation or other vegetation layers in this are just after the prediction of this maxent. We can't incorporate these vegetation or micro level data into maxent modeling, but we can do it that after the prediction, but after the prediction we will get an map. No, we will get an output from the maxent. So we can use that output in the QGIS software, and we can overlay the layers, and we can fine tune the predicted habitats, and also after that the ground truthing will support us to again fine tune this predicted habitat suitable.
Okay, but in the rainforest condition, the species are competing each other. In natural condition, how they are growing, how they are phenological, but in our treatment condition, that result will not get in natural condition, in control condition. The treatment condition is totally different.
Sir, we can do the acclimatization procedure even if we are germinating a species in the treatment condition. We can try for the acclimatization of this developed saplings in the natural habitat for 3 to 4 months, and after that we can plant these species in the identified, degraded, and the conservation areas. But another point I just want to add is that in our observation, the low elevation species like this Prioria, Diospyros is not having that much problem, even if we germinated these seedlings in our experimental setup or nurseries. But it is they can acclimatize, or they can actually that is their their actual niche is that the actual habitat is there. So they don't have we haven't observed much a problem in for those species to get acclimatized in that natural habitat.
Okay, fine. But some all species then not germinate in natural condition. They will need some treatment. So whether you applied the treatment, the same treatment which is found in nature.
Same treatment, yeah. That is I told earlier that is a interaction that is Diospyros plumnata and that their interaction with this Sileria. That we experimented here too, because we treated it with the fungicide and just for few hours, and this actually supported this collected seeds to break their seed coat, and which actually supported this seeds to germinate faster, and that one we have adopted. And another thing, we tried that if we are collecting a riparian species, we experimented or we developed such mimicry setup of their actual natural habitat. If it is a riparian species, we created a setup of a setup with water, sand, and everything, where the seeds were submerged in that. Likewise, we developed such kind of setups in our nursery, and these riparian species and as well as the low elevation species could able to germinate faster. And I can say one example in the case of one species, that is Humboldtia oleana. It is a riparian species, and Syzygium occidentale. All these are riparian species. They actually need high water content to germinate. So we experiment that kind of trials instead of any other chemical treatments or like that.
Okay, fine. Thank you. So you are not applying any other software for this modeling? You are not try for this. You do not try any other software for this modeling for this for your purpose of this work.
Sir, actually, I have done this for 25 species selected in my PhD work. Actually, that was based on different elevation from shola species, medium elevation, high elevation, and low elevation, and also riparian. And from different elevation, I have selected this species, and I have predicted this area and fine-tuned and refined the potential habitat for those species for their ecological restoration. It was 25 species study.
So which species are more grown in low level and high level altitude level?
Pardon? Which species?
Which species are more grown in low altitude and high altitude level? Which species are more grown in high altitude and low altitude level?
Sir, actually, the low altitude species, we can say that the low altitude species like
Riparian habitat, yes, riparian habitat. Riparian habitat, you mentioned a riparian habitat. That will be low level.
yeah, the riparian. In the case of riparian, we can say that Humboldtia oleana is a true riparian species, and other species like Syzygium occidentale, Garcinia whitey, Aprosa bordloni, all these are riparian species, and they can adapt to this riparian zones. And in the case of low elevation, we can say that the species like Diospyros, Prioria, Hypnocarpus, Dipterocarpus indicus, all these are low elevation species which can which we should plant in the low elevation, not in the high elevation. That I was trying to point out.
So why the particular species they are growing particular habitat? What is the reason behind it?
Species exist as a community, not as an individual. So in an ecosystem, there will be multiple species, but through phytosocial sampling, we can we can identify that which species is dominant, which is which is having much importance in that ecosystem, like that.
Ma'am, can you please note the question in the chat box?
Yeah, sure. One question, the use of remote sensing tools and tech-in sampling surveys and after action planting of species monitoring and identifying of suitable planting locations. Have you observed any need to do falling of trees in low level area? Yeah, the first question, the use of remote sensing tools in sampling surveys and after the action. No? The first thing, we use the remote sensing tool for the Maxent modeling and ecological niche modeling, and for fine tuning the predicted layer from the from the Maxent model, we can use the remote sensing tools, different vegetation layers, different elevation layer and all. So and after that, we need to do the ground truthing. We need to do the action in the direct field level implementation, and for identifying the suitable planting location to ensure that this is actually the location that we can plant for the species. That first, through the incorporating the vegetation layers, we will get a map, that where we can refine that area into which can be considered as a conservation area, which can be considered as a restoration area, and even we overlay with the converted habitats of our Western Ghats. We will get these are all the converted habitats of our terrain. So we can't plant our species there because there will be plantations, tea plantations, teak plantations, and all. So from that, we can fine tune which is the conservation area and which is the restoration area. But we can only ensure that through the ground truthing. For that, we need to identify the locations through the herbarium records, because the herbarium records will helps us to locate the actual distribution of that species. So from there, we need to identify and locate our targeted species, and we need to do the phytosocial sampling. And from there, we will get its associated species data. So if we get an area, predicted area, we need to go there and we need to fine tune where actually our plan can implement, or plan restoration can be implemented. Which is actually maybe in an identified area, we can see a rich evergreen forest and as well as a degraded site. So we can choose that degraded site, and from there we can initiate the ecological restoration process. And sir, any other questions?
I hope all the doubts are cleared, and ma'am, that was so informative session. As we come to the end of today's session, we extend our sincere thanks to Dr. Devika M Anilkumar for delivering such an insightful talk on ecological niche specificity and the conservation of threatened low elevation trees in the Western Ghats. Her session gave us a deeper understanding of biodiversity conservation, ecological research, and the importance of protecting threatened plant species and their habitats through scientific approaches and sustainable conservation practices. We also thank everyone who joined and actively participated in today's session. Your interest, questions, and engagement are what continue to make BioRex a growing and meaningful scientific community. Before we conclude, we would also like to remind you about tomorrow's BioRex Talk session on biodiversity conservation by Kavya N, final year MSc Botany student at Dr. Manmohan Singh Bengaluru City University. We encourage everyone to join and support our student knowledge sharing initiatives. With that, we officially conclude today's session. Thank you all for being part of this discussion. See you all at the next Biorex session. Thank you, ma'am.
Thank you.